Welcome back to the deep dive. If you've ever wondered what the absolute cutting edge of medicine looks like, well, today we have something that might just rewrite the entire drug discovery playbook.
We're talking about one of the biggest, if not the biggest, bottleneck in modern therapeutics, and that's finding new monoclonal antibodies, or MABs.
Right. And historically, finding one of these is just a physical, biological slog. I mean, you need a person or an animal that was actually exposed to the virus or whatever you're targeting.
Exposed to the antigen, yeah, the specific protein you want to neutralize. Then you have to screen, thousands of samples, just hoping you get lucky and find that one perfect antibody. It's incredibly slow.
And expensive. And you're always reacting to something that's already out there.
But what if you could just skip all of that? What if you could take the sequence of a new virus, plug it into a computer, and have it design a brand new, fully human, functional antibody from scratch?
And that is the exact promise of the research we're diving into today. The solution is called MAE, that's MAGE, the monoclonal antibody generator.
MG is, well, at its core, it's a specialized protein language model, a PLM. It's been fine-tuned to understand let's say, the intricate grammar of how to build a human antibody.
So our mission today is to give you the blueprint. We're going to unpack how MAGE pulled off this pretty remarkable feat of generating antibodies that are, get this, template-free, antigen-specific, and have the correctly paired heavy and light chains.
And against some really tough targets like SARS-CoV-2, RSV, and even the H5N1 bird flu, this is really about that shift from biology to pure computational design.
Okay, let's unpack that. I mean, we've had computational methods for a while now. What makes MAGE so fundamentally different?
It really comes down to two things: where it starts and what it actually outputs. Most of the older AI methods, they were really focused on optimization.
Optimization, meaning like taking something that already exists and just making it a little better.
Exactly. You'd give it an existing antibody, a template, and the AI would try to tweak it, improve its binding affinity. That's a process called affinity maturation. They were more like decorators than architects.
Huh, so they couldn't design the whole building from the ground up.
Not at all. And a lot of them were what we call structure-based. They needed these complex 3D maps of an antibody already stuck to its antigen to learn from.
And I'm guessing that data is pretty hard to come by.
It's extremely limited, especially for correctly paired human antibodies. MGE just sidesteps that entire problem.
Because it's purely sequence-based. It's just looking at the letters, the amino acids, not the final 3D shape.
Correct. It just reads the code. And this is the critical part. MGE generates the full paired heavy and light chains, the VH and VL.
And that pairing is non-negotiable for it to work right.
It's everything. An antibody is useless unless its heavy and light chains fit together perfectly to create that binding pocket, generating that successful pair from nothing. That's what makes ME a true first-in-class model.
So how did they do it? How do you teach a general computer model to speak fluent antibody?
They used a really powerful method called transfer learning. The foundation is a model called Progen 2.
Progen 2, okay.
It's a huge language model that was pre-trained on, get this, over 1 billion general protein sequences. So it already had this foundational grasp of, the physics and chemistry of proteins.
So it knew the basic language.
Yeah.
And then they gave it a specialized textbook just on antibodies.
That's a great way to put it. They fine-tuned it with a data set of 18,507 known antibody-antigen sequence pairs from all the major databases.
And the format of that data was key, I imagine.
Absolutely critical. They fed it the data in a very specific order. First, the antigen sequence, then a separator token, then the heavy chain sequence, another token, and finally the light chain.
So you're basically teaching it, when you see this problem, the antigen, I want you to produce this specific two-part answer.
That's precisely the logic. It's learning that conditional relationship. Now, here's a detail from the sources you can't ignore. Of those 18,000 pairs, a massive 67% were against coronavirus antigens.
Whoa, wait. 2/3 of its training was on COVID? Doesn't that risk? I mean, how do we know it's not just really, really good at making COVID antibodies and just sort of guessing for everything else?
That is the million-dollar question. Is MAGE a true generalist, or is it just a plagiarist?
All right, so that's the setup. AMEG is a new kind of AI that generates paired antibodies from scratch, trained on a big but very COVID-heavy data set.
Yep. So the first thing they had to do was prove the antibodies it was designing were actually new.
How do you even measure that? How do you prove novelty?
You quantify it with something called Levenshtein distance.
I've heard that term. It's It's like the number of edits of changes you'd need to make to turn one sequence into another,
Well,
right?
that's it, exactly. A spell checker for proteins, in a way. So they took every antibody MEG generated and measured its distance to the absolute closest sequence in the entire training database. A high distance means high novelty.
Okay, so what do the numbers say? Let's start with that high data target, the SARS-CoV-2 RBD.
They showed really significant novelty. The heavy chains, for instance, were on average almost 12 amino acids different from their nearest neighbor in the training set.
12 changes a lot.
It is, and crucially, the biggest differences were right where you'd want them. In the CDR3 regions, the hypervariable loops that actually make contact with the antigen. This was MG designing, not copying.
Okay, so they're novel, but the real test is, do they work? Do they actually bind?
They do. They synthesized and tested 20 of the designs against the RBD, and 9 of them showed confirmed specific binding.
Almost a 50% hit rate. That's pretty amazing.
It's very strong. And these weren't just weak binders. One of them, called RBD409, was a powerhouse. It showed highly potent neutralization of the virus with an IC50 of just 6.7 nanograms per milliliter.
Wow, that's therapeutic grade potency for something designed entirely on a computer.
Exactly. And it proved that MGE can find sequences that don't just bind, but bind functionally. And even the other binders were very novel. One had 18 differences from its closest relative and still bound strongly.
Okay, so that's the baseline. On a target it was heavily trained on, it produces novel, potent antibodies. But now we have to get back to that challenge we talked about before the break. Generalization.
This is where it gets really interesting. This is where you see the power of the language model itself. So they tested 2 other targets, RSV-A and H5N1.
Let's take RSV first. Respiratory syncytial virus. How much data did AVE have for that one?
Very little. The training set only had about 292 antibodies against the specific RSV protein they used as a prompt. A tiny, tiny fraction of the COVID data.
And the results?
Still incredibly robust. Out of 23 designs they tested, 7 of them bound. And again, with real potency. 2 of them, RSV-4314 and RSV-2245, had neutralization IC50s below 0.1 micrograms per milliliter. Very potent.
So it can handle sparse data. But what about no data, the H5N1 avian flu prompt? You said that wasn't in the training set at all.
That's right. This was the true zero-shot test. Could MAGE respond to a totally new emerging threat? They prompted it with the hemagglutinin protein from a recent H5N1 strain.
A sequence it had never ever seen before.
Correct. It had to generalize from what it knew about related H5N1 strains in the data, which were only about 91% identical.
So it's basically being asked to solve a problem it's never seen, just based on understanding the underlying principles. What happened?
It worked. 5 out of the 18 antibodies they made bound strongly. And all 5 of those were neutralizing.
Every single binder was functional.
Every single one. This is the critical result. It shows that MAGE could, in theory, be used to generate candidate therapeutics for a completely new pathogen in a matter of weeks, not years.
That is a fundamental change in how we think about pandemic response.
It is, but they wanted to go one level deeper. To really prove MAGE was coming up with new solutions, they needed to see the structure. So they used cryo-EM.
To get a 3D picture of how these new antibodies were binding.
Exactly, for 2 of the novel RSV antibodies. And what they found was fascinating.
Did they just bind to the usual spots?
Not at all. One of them, RSV-2245, did hit a known potent site called Site V, but the other one, RSV-3301, did something really unexpected. It targeted a place called Site I.
And what's special about Site I?
It's an epitope that people thought was mostly only accessible after the virus fuses with the cell, but this antibody hit it on the prefusion form of the protein.
Wait, so MG found a new way to attack the virus, a structural solution that years of biological screening had mostly missed.
It looks that way. It might be the first human antibody structure ever shown to hit that specific epitope on the prefusion protein. It demonstrates true creativity in the design.
That's incredible.
And the structure also proved something else, about something called somatic hypermutation, or SHM.
Can you just quickly define SHM for us?
Sure. It's the natural process your own immune system uses. After an antibody first binds something, your body makes tiny random mutations to the antibody's code to try and make it bind even tighter. It's like evolution in fast forward.
And MAGE's designs had these mutations built in from the start.
Yes, and that was the key. When they took that RSV-3301 antibody and reverted it back to its original unmutated germline sequence,
The binding got weaker.
it was substantially reduced. It proves the mutations MG introduced were absolutely critical for that strong binding. It's not just building a scaffold, it's mimicking the entire sophisticated refinement process of our immune system.
Okay, so MG can design novel, potent, and complex antibodies. But here's the practical question. Can you actually turn these into drugs? What about developability?
The crucial clinical hurdle, right? Are they stable? Are they easy to manufacture? So they checked this using an in silico tool called the Therapeutic Antibody Profiler, or TT.
And TP looks for what, exactly? Red flags for manufacturing.
Yep. Things like the length of the CDR loops, which can affect stability, or weird charge patches on the surface that can make the antibodies clump together. Things that are a nightmare for large-scale production.
And how do the MG antibodies score?
They passed with flying colors. All of the binding antibodies across all 3 targets scored well within the acceptable range you see for antibodies that are already in clinical trials. No major red flags.
Which suggests Meiji has somehow learned the unwritten rules of what makes a good, stable human antibody, even while it's creating totally new sequences.
It seems to have internalized those constraints, but, and this is an important limitation to state, the model is currently only conditioned to generate antibodies that bind.
Meaning, it's not trying to make the most potent one possible.
Not directly. It's not yet trained on functional data like maximizing binding affinity or neutralization potency. That still relies on making the candidates and testing them in the lab to find the best of the best.
So Meiji gives you an amazing pool of novel candidates to start from, which is the revolution here. It just collapses that initial discovery phase.
You've got it. It's that leap from laborious biological screening to rapid computational design.
So Meiji-E really is a paradigm shift. It's moving from finding antibodies in nature to truly generating them, creating these complex, paired human antibodies against targets the model has never even seen.
And the really profound implication here, which the source material touches on, is that as these data sets of antibodies get bigger and better, models like MAGE might eventually learn the general rules, the universal language that governs how any antibody binds to any antigen.
It wouldn't just be learning from examples, it would understand the fundamental principles of binding itself.
Exactly. And if it can truly generalize that knowledge? Well, here's the thought I want to leave you with. How quickly could future AI models shift therapeutic discovery from the months or years it takes now to maybe just weeks, even for an entirely novel pandemic-level pathogen? What does that world look like?
It changes the entire timeline of human survival. Thank you for joining us on this deep dive. We'll be back soon with more insights from the leading edge of research.